So just a little background on the habitat of this species. It was formed 25 million years ago in the Oligocene epoch of the Paleogene period. When it references austere terrestrial environments, it's just really extreme, harsh, really deprived of resources. They call it arid, hyper-arid, low rainfall, low humidity, high winds, and high ambient temperatures. The Arabian oryx, the study species, was thought to have ranged through most deserts in the Arabian Peninsula and Mesopotamia in the past, but there's only one self-sustaining population, and that's found in the Mahazat Said reservation in Taif, Saudi Arabia. They're among three endangered species, large ungulate species, ungulate meaning like hooved mammals. They found that these can survive long periods of drought lasting 4 to 6 months and the high ambient temperatures of this environment. There's brief periods of rain in the winter and spring, which is when the oryx has access to food and vegetation. But throughout the summer and the winter, the food content slowly gets depleted, and that's when everything gets scarce. They stated that they could survive indefinitely without access to drinking water. I'm not sure how they came to that conclusion that they could survive indefinitely, but previous studies had shown that their resting metabolic rate was reduced from 22 millijoules a day to 11 millijoules a day in the summer, and that their rate of water influx was reduced from 3.4 to 1.2 liters per day. So the purpose of this study was to investigate the mechanisms responsible for why the Arabian oryx has such high ability to acclimate to this environment. They also noted that this is a non-domesticated animal, so it's definitely a wild species, and it's desert adapted. So they predicted that the oryx would have evolved the ability to adjust their physiology depending on the resource abundance. This could be a decrease in the resting metabolic rate, which can be measured by a decrease in the thyroid and leptin hormone concentration within the plasma. A decrease in TWL, which is the total evaporative water loss. They can also test the concentration of the blood urine for homeostatic mechanisms used to cope with energy poor environments, or limited water environments, I'm sorry. Like these changing hormones could be glucocarotenoids that promote glucogenesis, which is basically using an energy source other than carbohydrates like fat storage. They can decrease in the thyroid hormone production to reduce their metabolic rate, and they can decrease in leptin, a hormone that's responsible for neural and hormonal responses to starvation. So for their materials and methods, again, these topics I'll be reading here. This study was located in the National Wildlife Research Center in Taif, Saudi Arabia, where that reserve was located. It was done between April and August of 2003. 14 individuals were used in this experiment, which were randomly assigned to an experimental and a control group. And then they also reported that there was statistic similarity between the groups in body mass and tarsus length. I think tarsus length is the length of their ankle. I couldn't just figure out exactly what it was. All I could find was the tarsus bone. But I'm assuming it's just the length of their ankle. And then for the experimental treatment, they gradually reduced their food and water intake by 15% every 3 weeks. Until this reached the level that they calculated during the summer months where they had the lowest intake of food and water. So they measured metabolism and evaporative water loss. They measured the minimum of resting metabolic rate and evaporative water loss during the day, which was during their resting phase, to more accurately determine the resting metabolic rate. Prior to testing, they deprived them for food for 50 hours as well. in their experiment, they, although that's a little exaggerated, built a steel respiration chamber, which is where they measured oxygen consumption rates and then converted that into heat production. Or prior to testing, I should say that they remained in the chamber 5 to 6 hours just to eliminate any sources of error. And they also calculated the water loss by measuring the dew point of incoming and outgoing air from the chamber. They measured the parameters in the blood and urine. So they drew blood from the jugular vein. It was centrifuged for 15 minutes at 2,500 RPMs within 30 minutes of collecting it. Half the plasma was frozen at -70 degrees to determine the concentration of hormones, non-ester-5 fatty acids, and beta-OH butyrate. The remaining half was measured for concentration of total proteins, glucose, urea, creatinine within 2 hours of collection. Leptin concentrations in the plasma were also measured through a competitive enzyme immunoassay. And they also measured the osmotic pressure of plasma in urine and the concentration of the urea and creatine in urine from the bladder after each blood sample. Alright, for the water budget and food digestibility, this was performed at the end of the acclimation period, which was at the end of the experiment, the 5 months. They constructed a water budget and measured the digestibility of the hay that they were consuming. To do this, they were housed for 3 days in a wooden cage with multiple layers of mesh that could collect and separate both the feces and the urine. The urine fell into an aluminum pan which contained paraffin oil, and this was used to prevent any of the urine from evaporating. The hay and water were provided in the same way as the end of the acclimation experiment, so they continued to feed them the same way that they would in a scarce environment. They also measured total water intake, which was the preformed food water, oxidation water, or the metabolic water, and drinking water. And they also measured water loss from the feces and the urine. This was determined by the moisture of the hay from the changing mass of it drying. So they measured it when it was wet, and then they dried it, measured it again to determine how much moisture was in the hay itself. They estimated metabolic water by assuming that 0.028 milliliters of oxygenated water was produced 1 kilojoule of energy. For food digestion, gross energy intake was measured, and this was determined by a gross energy content through an adiabatic bomb calorimeter. Nitrogen was determined from a Kagel extraction. Neutral detergent fibers were determined by a Goring and Van South procedure. Acid detergent was determined after the pre-treatment with acetyl-trimethyl ammonium bromide and acid media. And crude cellulose was determined according to Harvitz. So they basically took the apparent digestion as a percentage, was 100 times the amount they digested, grams per day, times or over the amount they ingested. And this was just basically a measurement of how much that they absorbed from that food. Statistical analysis, just some of the methods they used. Making sure the data between treatments was similar. They used an ANOVA to determine the difference between the metabolic rate and the evaporated water loss. And they, again, used 0.05 research articles. We'll read results.
Okay, so the results of this experiment. First, I checked the body mass, and just a reminder, the control is the 7 individuals that were fed and given water a consistent amount over time.